The neuromuscular junction is the synapse between motor neurons and muscle fibers, which is crucial for voluntary muscle contraction. Early degeneration at neuromuscular junctions is a hallmark of diseases, like ALS, often occurring before motor neuron loss. Quantifying NMJ morphology, innervation and denervation provides critical insights into disease progression and therapeutic effects. At the neuromuscular junction, a motor neuron releases acetylcholine, which binds to receptors on the muscle fiber and triggers an action potential along its membrane, leading to muscle contraction. Multiplex immunofluorescence is used to study the neuromuscular junction in detail. In this study, gastrocnemius muscles of mice were collected, perfused, embedded in OCT, and cut into thin sections. Multiplex immunofluorescence was then performed to visualize presynaptic terminals, motor endplates, and innervating axons. Here, we show a low magnification view of the gastrocnemius muscle, along with a high magnification close-up of the neuromuscular junctions. The post-synaptic component is labeled with alpha-bungarotoxin, the presynaptic terminals with SV2A, and the axons with beta-3 tubulin. A fully automated segmentation method was used to quantify NMJ morphology from the multiplex immunofluorescent images. In our method, each fluorescent channel is segmented separately using local thresholding, and innervation then is calculated as the overlap between the SV2A and alpha-bungarotoxin segmentations. A broad set of morphological features from the presynaptic and postsynaptic components is then used to classify each neuromuscular junction. We validated this method in the RNLS8 TDP43 delta NLS mouse model of ALS, which shows progressive ALS-like pathology, including NMJ degeneration, muscle atrophy, brain atrophy, and neuroinflammation. 8 weeks after induction, Biospective's low-dox RNLS8 mouse model displays a pronounced reduction in neuromuscular junction innervation relative to TTA control mice. At the same time point, low-dox RNLS8 mice also show fewer axonal projections and less complex presynaptic branching than control mice. Grip strength testing confirmed that RNLS8 mice generate less muscle force than their control counterparts. Serial non-invasive CT imaging demonstrated a clear time-dependent loss of muscle volume in RNLS8 mice relative to controls. Electrophysiological recordings revealed diminished compound muscle action potentials in RNLS8 mice, indicating compromised neuromuscular function. In summary, low-dox RNLS8 mice show clear disruptions in neuromuscular junction innervation and presynaptic architecture. Our quantitative analysis of these changes is consistent with clinically relevant readouts, including muscle weakness, EMG abnormalities, and muscle atrophy. Contact us at info@biospective.com to learn more about the RNLS8 ALS model and our NMJ staining and analysis services.